Hi, everyone, and welcome into Conversations with Dr. Charles. I'm Pat Bryant, and I'm happy to be with you today with Dr. Charles Ackle, who you can see here. Today, we're going to talk about the second part, and we're not sure how many total parts there will be, right, Charles? I mean, this is such a rich conversation, but it's our second part in talking about the roots of Emmy. Where did Emmy come from? Where did this product come from? Let me introduce Dr. Charles for you very quickly. If you don't know Dr. Charles, he's the Chief Medical Officer here at Emmy. He's a fellow at the Royal College of Surgeons in the UK. You'll hear his fine British accent in just a moment. And he was awarded several prestigious prizes, including the Hallett Prize, and received a lot of awards for his undergraduate work as well. He's trained in medicine at Guy's Hospital in London, and he researched in transplantation and also immunology. And Charles, I think that lit a fire in you that still burns brightly today around this whole concept of the immune system and immunology, which we're going to get very deep into. Charles was a prominent London-based surgeon and also a pioneer in the development of laparoscopic surgery, and he's responsible for training many of the fine surgeons in the UK and Europe in laparoscopic surgery, and importantly, in establishing better relationships between private and public partnerships in medicine in the UK and Europe. So as a short introduction, Charles, that's who we're telling folks you are, and welcome in again. These are wonderful conversations.
Thank you very much, Pat. Very humbling and delighted to be here and to take on the next step.
Great. Well, as we dive in, we just want to remind you, if you'd like to receive information about these videos when they're available and new, be sure to hit the subscribe button below. We're excited for you to tie into that. And also, if you want more information about our product and some of the background that we're going to be talking about, be sure to go to emmy.co. That's emmy.co. And you'll get some great information there. So Charles, we're going to talk today. If you didn't watch the first video about Emmy and its background, please go do that. You're going to miss rich stories that you were getting into. And I want to pick up where we stopped off on the last one, which is you were talking about the ties between immunology and the immune system and cancer, and the way that transplantations, there were some interesting issues that were occurring with transplantations, and then tie that over into the bacteria strains that we're talking about here today that ended up being found in Emmy.
Good. Yes. I was telling the story about the kidney transplants and cancer, which you'll pick up in the previous episode. Just to reset, if you like, another story is I was talking about malignant melanoma, a very immunologically active cancer, and it was one of the ones that really pushed us into recognizing how important immunology is in terms of trying to treat cancer. Something that really, even as long ago as 2010, people just didn't get. And I'm talking about doctors, never mind patients. Patients got it. The doctors have always been very slow, unfortunately. But one of the cases, for example, is a lady who was pregnant and had malignant melanoma, and the baby was infected, if you like, or affected by malignant melanoma whilst in the uterus. The cells crossed into the placenta into the baby and infected the baby. When the baby was born, it had malignant melanoma. As soon as the baby's immune system kicked in over the period of 2-3 months, the melanoma disappeared.
Amazing. Wow.
Amazing, amazing. And the reason is that, of course, the mother has a degree of immunosuppression, as the baby has, because when the fetus and the mother interact, there has to be something that stops the mother rejecting the baby, and equally, the baby rejecting the mother. And we have issues, rhesus factor, you've probably heard about rhesus factor disease, is one of those situations where the immune system goes wrong and the mother attacks the baby. And that's really an important story. But the cellular reaction is quite extraordinary. And the baby, once its immune system kicked in, it was able to deal with the cancer. So that really gives you the experimental model that you couldn't possibly do in a human except by observation, right? Well, we knew about this, and I told you the background of Mycobacterium vaccae and its effect as an immunoadjuvant. It was then developed into a cancer agent because that was the recognition that this might have an effect in cancer by boosting the immune system, by triggering responses, danger responses, which would then attack the cancer. And various trials were done. We didn't know how to do it properly in those days, or my predecessors didn't. And one of the things we didn't know and learned later was that you've got to keep giving the material. You can't just give one dose. You've got to keep giving it.
And why is that, Charles? What's going on in the body that's causing that to be true, as opposed to like a prebiotic or things like that, that we hear about?
Well, the thing is that you need constant stimulation. There's a constant interaction, a to and fro between you and your environment, between your guts bacteria, your skin bacteria, your lung bacteria. There is an interaction that goes backwards and forwards. If that interaction becomes stale, you get what's called tolerance. And this is where cancer, for example, is a master player. It induces tolerance in the immune system, which means that the cancer does not become recognized by the body. So to break tolerance, you need to keep hitting it and waking it up. Hey, wake up, wake up, wake up, wake up, wake up. Come on, pay attention. And that way you get a better response. The systems in biology, as in elsewhere, tend to just dampen down and shut down. Cancer will induce the production of what are called regulatory T cells, among other cells that will dampen down the immune response. So you've got to keep hitting it, and that's why the repeated dosing is so important. Well, in a lung cancer study that we did, which showed quite a good benefit in a subgroup, but only in those who had repetitive doses, then we learned that actually you've got to keep giving this, and we've modified accordingly, and that research is going on, but using a sister organism called Mycobacterium obuense, Obuense, which is from the prefecture of Obu in Japan. It and aurum and vaccae were all discovered by Professor Tsukamura, who he and his wife used to travel all over the planet and collect interesting organisms, particularly mycobacteria. Some people collect stamps, some people collect cars, some people collect. Tsukamura collected
Usual collection.
Yeah, so he collected these, and we and John Stanford looked at this and recognized that both obuense and aurum were very, very close and would behave quite well. And for various reasons, we did most of the work on obuense, and we put aurum to one side.
So, Charles, let me ask you a question there before you move off of the aurum piece. There are multiple strains of Myceliaceae bacteria that are in that same classification, but not all are beneficial, correct?
We don't know. The truth is, all of those are by definition, because they're Myceliaceae bacteria, non-toxic and non-pathogenic. We know from broad studies, because we've collected, I'm afraid we've worse than Tsukamura. We've been all over the planet and collected stuff from the Atacama Desert to the Himalayas. We've got bacteria from all over the place. And it costs a lot of money to analyze and work it out. But we think it's essentially spectral changes, that it's changes of degree rather than absolute changes of pathogenicity. So that's why we think aurum will be and has effects, and we've proved with quite a lot of science that actually it produces very similar signals to the other two organisms. In every way, it's almost identical. The problem with pharmaceutical grade development is it costs millions, hundreds of millions of dollars to do it. It's thought $2 billion now to bring a new cancer drug to market. It's fantastic. And the cost of these drugs is $100,000 a throw. It's unsustainable. And that was part of our ethos in the TB world when we wanted to do it at $1 a day, where a sub-Saharan African could afford it. And if you're not insured, you're tight on money. We want people to have a benefit. But we've done the development with obuense and with vaccae. And one of the things that came through, and there are two stories. One is that in the lung cancer cases, and in fact, in all the cancer cases that we were studying, the nurses noted, as did the patients, that those patients who were taking the mycobacteria seemed to be able to cope with the diagnosis of cancer and their pain and the disability far better than the others. And that was shown in a paper published from the Royal Marsden, which is our top cancer hospital in the UK. And that prompted neuroscientists, and one of the prominent ones is Professor Chris Lowry at the University of Boulder in Colorado, to look at why this might be. And he was the first person to describe the fact that when you give an experimental animal vacci or obuensi, or indeed if you give it aurum, you will stimulate neurological changes that allow the patient to deal with stress, improve coping behavior, and this is extraordinary. So if nothing else, we have a quality of life benefit for those cancer patients. It helps them to cope well with this awful disease, even if we don't necessarily cure them. So aurum comes from that background. And interestingly, it was Sir Christopher Gent, who is a very well-known businessman. He was chairman of one of the big pharmaceutical companies here. And I remember talking to him. And he said to me, look, why don't you do this as an oral material so that lots of people can benefit, because the injections cause a scar, there are all sorts of issues. So we chose Mycobacterium, Mycolicibacterium aurum, and you'll see it's called Aogashima because Tsukumura found this on the island of Aogashima, which is a beautiful volcanic island, just again off the prefecture of Obu in Japan.
Yeah, it looks like a blue rim volcano. It's just gorgeous.
Check it out. It's the most beautiful. You'll see pictures on the net. It's gorgeous and volcanic. But its aurum versions are present in soil over probably 30% of the planet. So
but
they're
not commercially available everywhere, correct?
No. And
the difference to have it in your soil, it's another one to be able to mine that, I guess, for lack of a better way to say it.
And conform to strict regulatory processes, which we do. It's essential that we don't have what you Americans would call BS, if you'll pardon the expression. Yes. We do the things properly, and or at least we try. And so each particular version of aurum will need a particular assessment. So we've done all the toxicology. We've done a lot of work on aurum to make sure that it's safe for human consumption. And that's why we have it available now, and it's at a cost that is very cheap. Now, if we start doing detailed clinical trials at clinical grade, we're going to spend hundreds of millions of dollars, and I'd rather concentrate that money on the sister organisms because we've got a lot more background data and try and get that through to the cancer patients.
Yeah, go ahead, Charles.
So the aurum, the whole point about why it works is the difference is why does it work orally when the other one is by injection? Mm-hmm. And the answer is actually they both work orally. And that is because there is a specific set of immune cells in the gut, which is the biggest immune organ in the body by a mile, which will specifically pick up certain organisms, including Mycobacterium, Mycolicibacteria. You need tiny amounts, minuscule amounts. And by the way, these bacteria are pooped out. They don't thrive. They don't colonize the gut. They're not a probiotic or whatever you want to call it. They're not like lactobacilli or bifidobacter or any of these other standard microbiome organisms. It just goes through, but it's detected in tiny amounts on these cells, which then talk to the immune system, and they then talk to the brain. What's absolutely incredible is the way that the brain interacts with the gut. And we now know, just in the last year, from papers published in Nature and other big scientific journals, that the brain can actually affect the gut and can inform the gut immune cells exactly what things to react to. This is mind-blowing. I mean, it's really fantastic stuff. Yeah.
And it's what we read about, you hear a lot about the gut-brain axis, but I don't think there's a lot of people who fully understand it. We're actually going to talk about that in a future conversation.
We'll go into detail. Yeah.
Yeah. I do want to ask you before we run out of time, why does the immune system need what you've called an orchestrator or a conductor. Why is that important?
Oh, first of all, we think we now realize the microbiome, which is all these bugs that live in, on, and around you, is absolutely fundamental to your responses for almost everything. And in particular, by the way, cancer, but allergic responses are very relevant to the way your microbiome works. And on a future occasion, we'll talk about the urban rural split. But these organisms are absolutely fundamental because you probably have 40, 50,000 different bacteria in your gut. I mean, these people that try and sell you 5 bacteria or whatever it is, they're dreaming. You've got 40,000 to play with. Have you seen an immunology book by any chance? I mean,
No, I have not.
well, don't, because you won't be able to lift it off the ground. The number of different cells is mind-boggling. And it's exactly like an orchestra. You've got so many instruments. How on earth, if they're all playing music at the same time, how on earth can you make beautiful music? I mean, it doesn't make any sense. Something has to orchestrate, something has to coordinate all these things. Of course, it's the brain, probably, but
Mm-hmm.
it needs adjuvant. And I've mentioned about adjuvant in a previous talk, the critical importance of a broker who can handle all these things. And one of these adjuvants we know are the mycolysibacteria, these environmental bacteria that constantly need to keep things under control. And that's where we feel IMI is very important. We're replacing something that has been lost in the natural environment, which every human being, probably every animal, needs. In the wild, animals go to salt licks. You've probably seen mud licks in
Yes.
parts. We go and lick the mud, they eat the mud, and that. I mean, why would they do that? Pilgrims go to this church in Chimayo in New Mexico. They eat the mud from the church because it's supposed to have miraculous properties.
Mm.
Um, Children who are iron deficient will eat soil. So there is a natural tendency for all of these things, and there's a lesson there to be learned. And we think IMI is a major element, or organisms, of course, like IMI, are critical.
Absolutely. And I've been so impressed as I've learned so much more about Emmy and the roots of Emmy, the concept of democratizing the product. I think it's a wonderful concept to say, you could have taken it a pharmaceutical route, but you wanted to make it available to people. And while we may not be able to make dramatic claims that would have to be proved out by expenditures of a lot of money, we do know that there are proven elements, as you talked about with the gut-brain axis, which we're going to get into in more depth. But for today, or at least for this session, Dr. Charles, we have run out of time, and it's just amazing to me how fast the time passes when we start getting into this. And I'm enthralled, even though I've heard some of these stories once. As I hear them again, they're just so rich. So thank you for offering that. If you've enjoyed this, you're watching this, we're going to do this again and again and again. So be sure to tune in. If you subscribe, if you hit that subscribe bell below, what you'll find is you'll get notifications on when we've posted some of these new ones. We're going to continue to weave in Emmy and how it fits with some of the things that we're going to talk about. But if you're interested in more depth on the microbiome or stress, stress resiliency, the gut-brain axis, and a few other things, we're going to talk about those. So be sure you come back and visit with us again. Our product is called Emmy. And you will find more information about it if you go to emmy.co. That's emmy.co. So again, Dr. Ackle, thank you so much, and I'll look forward to our next conversation.